In this video, we're going to be talking about the second part of photosynthesis, the carbon fixation part. Last time we talked about the light-dependent reactions, which is where the energy comes in. It's where it's captured by the sun and converted into a form of chemical energy that the chloroplast can then use to fix carbon. So the light-dependent reactions occur on the surface of the thylakoid membrane, and when they're all completed, we have an input of ATP and an input of NADPH, which all go into the carbon cycle. And so today we're going to talk about the carbon cycle. How does carbon dioxide get captured from the atmosphere and become glucose? Okay, so the Calvin cycle. There's a lot of nitty-gritty details to the Calvin cycle, and so this might take a little bit of time, but go ahead and take your time, take careful notes, and pause the video if you need to. So I'm not going to try to keep track of every single atom in the Calvin cycle. The atoms that I'm going to care about for the Calvin cycle are carbon atoms. We're going to count carbons, and we want to make sure that we're keeping track of all of the carbons. But all of the oxygens and hydrogens are also being shuffled around during some of these steps, and so I don't want you to worry about keeping perfect score for them, especially the oxygens, which have various things that happen to them. Okay, so the Calvin cycle starts with an input of carbon dioxide. Carbon dioxide, of course, has one single carbon. This carbon is going to come in and it's going to join to a pre-existing sugar molecule. Remember, all this is taking place in the stroma of the chloroplast, so all of these different molecules and all of the different enzymes involved in all of these steps are just crashing around inside the stroma, and they collide with each other by chance frequently enough that this happens, all these processes happen very smoothly. But these are just in the stroma floating around. So floating around in the stroma are these 5 carbon sugars called RUBP. Make that a little smaller. 1, 2, 3, 4, 5 carbons strung in a chain. And like every monosaccharide, there are also oxygens and hydrogens hanging off of these carbons in various ways. This is a linear sugar, which means that instead of being joined together into a hexagon or a pentagon, it's just a single line, but it's still a carbohydrate, it's still a monosaccharide, RUBP. Okay, so the 1 carbon of carbon dioxide and the 5 carbons of RUBP joined together. They're joined together by an enzyme called rubisco, one of the hardest working enzymes on the planet. And rubisco joins together 1 carbon here and 5 carbons here to make 2 3-carbon things. That's actually what rubisco winds up making, is 2 3-carbon sugars. I'm not going to give these guys a name because they get transformed pretty quickly. Okay, so we've got a total of 6 carbons here and a total of 6 carbons here. These 3-carbon sugars are not yet ready to be made into glucose. They need some transformations. They also need to have some additional hydrogens added to them. So they're going to go through a series of stages. And along the way, on this series of transformations, they're going to be transformed from a 3-carbon sugar that looks one particular way to a different 3-carbon sugar. And these steps along the way take energy. So this is where our ATP and our NADPH come in. So you add 1 ATP per sugar, and we also add DP plus. We also add our NADPHs along this stage. So we add in an input of energy and we add in an input of hydrogen in order to change these 3 carbon sugars to these other 3 carbon sugars. And these 3 carbon sugars, these outputs, are called G3P. So we've made 2 G3Ps, and G3Ps are the building blocks of glucose. If you take 2 G3Ps and you put them together in the right way, you make a glucose molecule. And that's great. Doesn't it seem like we should just stop there? Well, no, we're not going to stop there, because where did this RUBP come from in the first place? We can't just have an infinite supply of RUBP. It's already a sugar, so it had to come from somewhere. So the Calvin cycle, the cycling part of the Calvin cycle, is the part that regenerates this RUBP. So we're not just going to have an input of 1 carbon dioxide. We're going to have an input of 3 carbon dioxides. We're going to add those 3 carbon dioxides to 3 RUBPs, and we get not 2, but 6 3 carbon sugars, and not 2, but 6 G3Ps. So we've got sort of 3 turns, or 3 half turns of the cycle. Let's make sure that everything is accounted for. We have 3 times 5 is 15, plus 3 is 18 carbons going in, and we have 3 times 6 is 18 carbons coming out. Great, haven't lost any carbons, that's what we care about. Now, at this point when we have 6 G3Ps, once we have made 3 half turns of the cycle, then 1 of these G3Ps is going to leave the Calvin cycle. This 1 G3P is going to go on to become a glucose. So it's going to join together with another G3P, and together they're going to make a glucose, which you know is 6 carbons. So really it takes 6 turns of the cycle to make a single glucose. Every 3 turns, 1 G3P leaves. It joins together with another G3P, and you get glucose out of it. This leaves 5 remaining G3Ps. So these So these 5 G3Ps, we're going to regenerate our RUBPs from them. So this is 5 times 3 is 15 carbons, and RUBP is 3 times 5, or 15 carbons. So through a series of steps, we're going to transform the G3Ps back into RUBP. And this stage actually also requires an input of energy. So it requires 3 ATPs to create 3 RUBPs back. And now what have we done? The cycle turns 3 times to make 1 G3P, 3 more times to make a second G3P. Those 2 G3Ps join together and make glucose. Phew. All right, so that brings us to the end of photosynthesis. This is the overall equation for photosynthesis, which is that 6 waters plus 6 carbon dioxides yield a glucose and 6 oxygens. This is universally true in all photosynthetic systems, but this process is not perfect. Just like any biological process, it's a little bit lossy. So what I mean by that is that we don't always get perfect yield. Sometimes the plant has to work a little bit harder or produce a little bit more ATP than it might like because some of that energy gets lost. Processes are imperfect, and biological processes are particularly imperfect. Plants have developed 2 really interesting modifications on the Calvin cycle that let them improve their photosynthetic output, that make them produce a little bit more glucose than they might otherwise. So modification number 1 is a modification that desert plants do. So if you are a plant in the desert, then your major concern is holding on to water. You don't want to let your water dry up. But the problem is, every time that you're breathing in carbon dioxide, anytime the stomata, those little pores on the underside of a leaf, are open, that allows water to escape from the plant. So as long as carbon dioxide is coming in, water is leaving. And that's bad. So what these desert plants do is they actually separate out the light reactions from the Calvin cycle in time. What they do is they run all of their light reactions during the day, and light reactions don't require any carbon dioxide. So they run all the light reactions during the day, they store all of that energy in a different format, ready to use later, and then at night, they can open up their stomata when it's cool out and they're less likely to lose water through evaporation, and then perform the Calvin cycle, which is also sometimes called the dark reaction because it doesn't require light. They perform the Calvin cycle at night. So that's pretty cool. The other modification on the Calvin cycle is one where that takes place right at that first step when carbon dioxide is joining with RUBP by that enzyme Rubisco. Rubisco is a really hard-working enzyme and extremely common on the planet because it's present in all green leaves of all plants. But it's actually kind of a bad enzyme in that Rubisco is really error-prone. It tends to make a lot of mistakes, and it tends to make a lot more mistakes when oxygen is present. And so the second modification that some kinds of plants have created is to take Rubisco and move it away from all of this oxygen that's being created. Actually, to physically put the Rubisco in a different part of the cell and to move components around the cell differently in a way that isolates Rubisco from oxygen. And so that's also really exciting. And that modification was came about in grasses. And grasses are extremely successful photosynthesizers because they're much more efficient than plants like trees are. Which means that bamboo is a type of grass, and bamboo is the fastest growing plant on the planet. It can grow 1 meter in a day, which is shocking. It's almost fast enough that you can see it growing, which is crazy. So next time we're going to be talking about how plants and also animals and also yourself use glucose in order to extract its energy. So next time we're going to be talking about the mitochondria. How do we get the energy that we put into this glucose? How do we get it back out?